The Life Sciences DNA podcast is sponsored by Agilicium Labs, a collaborative space where Agilicium works with its clients to co-develop and incubate POCs, products, and solutions. To learn how Agilicium Labs can use the power of its generative AI for life sciences analytics, visit them at labs.agilicium.com. Amar, we've got Mohan Uttarwar on the show today. Who is Mohan?
Mohan is a biotech entrepreneur who is the co-founder and CEO of OneCell.ai. He's a repeat founder with deep experience in both diagnostics and therapeutics. And as an innovator and investor, he's got more than 30 years of experience in life sciences, precision diagnostics, and healthcare technology.
And what is OneCell.ai?
OneCell.ai is a precision oncology company that's using AI and single-cell analytics to help pharmaceutical teams accelerate the next generation of cancer diagnostics and therapeutics. Its platform provides real-time tumor profile that can help drug developers make appropriate patient selection, accelerate development, and provide more nuanced understanding of a patient's response to therapy. But at the same time, this platform can be used to provide earlier diagnosis and guide personalized treatments by the oncologists.
And what are you hoping to hear from Mohan today?
I want to understand how OneCell.ai's platform works and the potential for real-time cell profiling to reshape clinical trials, drug development, and diagnostics.
Before we begin, I want to remind our audience that they can stay up on the latest episodes of Life Sciences DNA by hitting the subscribe button. If you enjoy the content, be sure to hit the like button and let us know your thoughts in the comments section. And don't forget to listen to us on the go by downloading an audio-only version of the show from your preferred podcast platform. With that, let's welcome Mohan to the show.
Mohan, thanks for joining us. We're going to talk today about how AI is transforming both cancer care and the development of new therapies, OneCell.ai, and how its liquid biopsy diagnostics are used by both clinicians and researchers. So let's start with the liquid biopsies. For audience members who may not be familiar with these, can you explain what they mean and how they work?
Thank you, Amar. Thank you for inviting me here. It's a pleasure and appreciate an opportunity to talk a little bit about the field as well as what we do, why we are excited about what we do at OneCell. Cancer is a deadly disease, we all know. One shoe doesn't fit all. So personalization of therapy is the only way cancer can be managed. This whole field has evolved into what they call precision oncology, which refers to personalization. Historically, what happens is somehow, you find something, then the doctor tells you go into the imaging, right? You do the radiologic scan, and then radiology finds something, but then the pathology is called for. Which is the process of taking a biopsy. Biopsy is the process of taking a small tissue sample from the area where you suspect that there is cancer and further analyzing it. It's sent to the lab, that tissue sample. It's called tissue biopsy. Tissue sample is sent to the lab. The lab does the processing of the sample. A glass slide is created, okay? And a pathologist puts it under the microscope, does the reading, and based on the pattern he or she sees, they write a report. And based on that report, a treatment is given. Okay. This process is called tissue biopsy. Okay. Now, tissue is an issue because it's a surgical procedure. It's expensive and it's not an easy thing to do.
Yeah, okay? And it's invasive, right?
So that's invasive, right. Yeah. It's invasive. Yeah. It's this thing. As the field evolved and the science evolved, the information that you get from tissue, similar information you can get it from blood, liquid. Okay, so that's why Dr. Klaus Pantel and Katerina Penneberes in Europe, they coined this term liquid biopsy. That means you take a simple blood draw, which is non-invasive or minimally invasive, and then you process it, in the lab and give a similar report. So that's the liquid biopsy aspect, right? From the sample of blood
Mhm,
draw that you
mhm,
create the reports, generate the reports
mhm.
using next generation sequencing.
Okay. Now,
Okay.
when you're doing the liquid biopsy, like, is this circulating like tumor cells or circulating tumor DNA? Are they available for pretty much most cancers, or is that like more prevalent in some cancers versus not, or even like the staging, like maybe like when it goes metastatic, it's more present. Like, like how does that work?
What happens is cells die, some natural death and tumor cells, right? So when they die, it spits out the DNA. That's the circulating DNA or cell-free DNA. Okay. Subset of the cell-free DNA, right, from the dead cells. If it is a tumor cells, the death of a tumor cells, which spits out the DNA, it's called ctDNA. Okay. Information that you generate from the ctDNA, circulating tumor DNA, is most relevant. Okay. But the problem with the liquid biopsy from the blood based stuff is it's very difficult to figure out whether it was a cell free DNA from a normal cell or versus the dead tumor cells, right? But now with advent of AI and analysis, right? You know, people have been able to figure out the difference. Okay, and it's a good biomarker. It tells you that more the ctDNA, the more the prevalence of cancer. So you map it, right, and you have the hypothesis.
Okay,
Okay.
so it's like the higher stage, greater stage you are, it's likely to have more
Correct.
ctDNA. Okay, and is it like in pretty much most cancers you see that, or like some cancers more than the others?
Yeah, some cancers more than the others, but most
Mhm.
cancer would have some DNA,
Okay,
ctDNA, right?
okay,
And we're referring to tumor, I mean, the
yeah,
solid tumors,
yeah,
right?
yeah.
Okay. So tissue biopsy, the next generation was liquid biopsy, and liquid biopsy is referring to ctDNA. However, it is not a full representation of a tumor inside the body of a cancer patient. Okay. These are essentially DNA fragments coming out of the dead cells. Okay. So, what we are doing now is taking that industry forward to the third generation. Okay. We call cell biopsy. Okay. Which is basically a next generation of liquid biopsy, but coming out of the circulating tumor cells. Now, let me explain the CTC or circulating tumor cells. A gram of tumor, okay, sheds about 1 million cancer cells in the body, in the bloodstreams. So that may seem a lot, but we have trillions of cells in our body. So that means it's 1 in 1 billion circulating tumor cell. It's like a needle in the haystack. Okay. All right. Now, however, these are the real culprits. So what happens is mostly 95% of the cancer deaths happens because of the reoccurrence. Okay. The root cause of the reoccurrence, okay, is metastasis, a process where cancer spread from its origin. It might be breast cancer, but because of the circulating tumor cells, the cells that are shed, they go run around. Most of them die. Most of them are killed by our immune system, but some strong ones evade the immune system and they settle somewhere else. Okay, so they find the fertile ground. Maybe it's lung, maybe it's brain. Somewhere they go and settle down, and the secondary tumor grows. Okay, okay. So these are like the fugitives on the run, but they are the microcosms of creating secondary tumor cells. Okay, and that's the root cause. Okay, so historically, it was very difficult to find those CTCs, those 1 in 1 billion circulating tumor cells. So, okay, and so But now the new science, the groundbreaking science, to capture that 1 in a billion circulating tumor cell, okay, at a very high degree of sensitivity, high degree of precision, the purity of capture, okay, and the high degree of simplicity and lower cost. Okay, so why we are excited about this whole field is because one cell has this groundbreaking science. And if I take these three axes, order of magnitude higher sensitivity, order of a magnitude higher purity of capture, order of a magnitude lower cost, it's a
Yeah.
1,000 times better price performance than anything that existed before.
Yeah.
Okay.
And it seems like so then that's going to be both from like the technical aspect as well as like the algorithmic or AI aspect, right? Like are
Yes.
these like the both aspects
Yeah.
going
Yeah.
here like to make this
Sure.
better?
Sure. Sure. Second generation of precision oncology also was, about multidimensional data, multimodal data, multi-omics data. Cancer is like an elephant and a six blind man touching it and describing what it is. A genomics is a science of genetics, right? It tells you something very important, but it doesn't give you a full picture. Just like a blind person touching, the leg of an elephant, if you ask him to describe, he'll say, oh, elephant is like a leg. Another blind person touches, let's say the tail, and it says, oh, it's like a snake. So unfortunately, the genomics, the transcriptomics, the proteomics, the metabolomics, the metabolomics, these are all fields of science, but they are looking at cancer into the silos with their own lenses. Unfortunately, cancer, everything, this has to be all integrated. So the first generation of precision oncology was driven by tissue biopsy and the genomics data. Companies like Foundation, they did very, very well. They established as the leader. But tissue is an issue. So second generation, the liquid biopsy, right, which is the blood-based stuff, right, that came about. But what has been not fully equipped is, again, that was really focusing again, on the genomics and, the transcriptomics. But the DNAs, they don't, cannot get you the transcriptomics or maybe a little bit, but definitely not proteomics. Okay. So the third generation, which we call the cell biopsy, has the ability to do all three pieces together: the genomics, the transcriptomics, and the proteomics. Now, what happens is multi-omics, it's a big data problem. Cancer, as much as it's a disease, it's also a data problem. And the data is overwhelming. So this is where AI comes in handy.
Even
Analyzing this multidimensional, multimodal, multi-omics data, making sense out of it, okay,
Yeah.
correlating the data and predicting the outcome is the key challenge, right?
So the
Yeah.
challenge here I see is that like usually like when you do like genomics, proteomics experiments, right? Like you have like a normal tissue, you have a disease tumor tissue, right? And you're looking at like differences here. But then what you're collecting, that has like majority of that is already normal anyway. So how do you then detect the specific cancer DNA or the cancer proteins from that? Because that's a much higher level challenge than what people usually do, right? So how do
Absolutely.
you do that with the AI?
Yeah. Very, very good point. Excellent point. In liquid biopsy, I told you it's all about DNA, and you have a challenge because you start with a noisy data. You don't know whether that DNA came from a normal cell, dead cells, or a circulating tumor. I mean, the ctDNA, right? Yeah. And so when you start with the noisy data, again, you have to apply AI and deep analytics, more resources to basically, denoise that data. Okay. The next generation technologies, particularly the one that we have invented, you are already clean. You're starting with a cleaner data because we have figured out capturing that 1 in a billion circulating tumor cells.
Okay.
So we already know that our starting point is a tumor cell.
Oh, okay.
Okay.
So you have the technology to then detect the tumor
Correct.
cell
Correct.
specifically.
Correct.
Okay.
Okay.
Now, does that depend on like the kind of like surface markers there are on like the tumor cell, or
Very good point. So CTC is not new. It's been there 10 years ago. Okay. Unfortunately, what happened was the sensitivity wasn't there. And the endpoint was you capture the circulating tumor cells, you count and report them. Okay. So the clinical utility was limited. It told us that there is a problem. Okay. Your PET or CT scan or MRI might be clean, but if you find CTCs, that means, there are some problem somewhere. Okay. So the scientists would say, oh, we found the problem. Okay. But the doctors would say, so what? My patient is happy because the PET is clean. And now you're telling me you found a CTC and you can't tell me what to do about it. Okay. So it's a very classic dilemma. Scientists get excited with what if. Okay. But the doctors are saying, so what? What if I found a CTC and the doctor is saying, so what? Unless you tell me what is the actionability. And and the science stopped there in the past. But today, with the advent of next generation sequencing and with AI, okay, we are able to analyze the information in that single cell. Okay, the genomic data, the transcriptomic data, the proteomic data, and tell the doctor, hey, it may be the cell coming from lung. You did a PET on basic breast, which was the original stuff, right? So without AI analyzing the cell information that is hidden in that circulating tumor cell, we could not enable the doctor to take those critical clinical decisions. Okay. So it's really the integration of information from the biology of the cell, the science part, and the AI working on analyzing the genomic data, the transcriptomic data, the proteomic data, right? So, like you said, cell surface marker. We know historically that if it's, let's say, HER2 marker on the surface of the cell, very likely that cell must be coming from breast cancer site. If it's an EGFR, very likely, again, no guarantees, but very likely it's coming from a lung cancer, right? Also, the way morphology of those cells look is very unique. So we use AI to analyze, how the cell looks, the morphology, the size, the shape,
okay?
the density, the contrast, right? So there's
So
a
there's
lot
the
of
imaging as well then.
image analysis of the
Okay.
cell that we
Okay.
capture, right?
Okay. So there's in addition to the multi-omics, you're also doing the image analytics.
Absolutely,
Gotcha.
absolutely.
Okay.
Even before we do multi-omics, we
Okay.
do the analysis of these cells, right?
Okay.
We do the characterization using our AI algorithms,
Uh-huh.
and we give enough information. Now, one other, fact that is actually very revealing is that many times the CTCs are individual CTCs, but many times you have this, what we call the the combinations, right? You have the group of cells hanging together. Okay, now the risk profile of those patients prognostic is much different. It's much higher risk. So, okay, so again, imaging reveals those kind of things. Okay. So there's interesting stuff, right? Now, how do we capture these circulating tumor cells, right? The CTCs, there are two methods for capturing. One is size based, right? So tumor cells are usually larger in size, and there are techniques, microfiltration. There are techniques to really do the nano fluidics to capture those cells. That's one method. The other method is called affinity based. So tumor cells have certain types of proteins on the surface of these cells. EpCAM being one very popular protein that is on all solid tumors that are epithelial in nature. Okay. A transferrin in another protein that is expressed. Okay. So what happens in the size-based capture? The cells go through a lot of, uh, pushing and pulling, nanofluidics. You're forcing this, nanofiltration. You're pushing it. As is, when cell leaves the tumor, okay, uh, it was in a very comfortable position sitting in the home, and now it has to fight the whole battle, right? Immune system really is, the aggressor which is trying to kill it. So only a few remain. They, you know avoid all the forces against them, and so they are right, but they go through a lot of, fights. They go through a lot of transitions, transformation, right? Pressure, right? So as a result, size-based capture has not yielded great results. Okay. Affinity capture relatively is less stressful on the cells. Okay. So that's been historic perspective. The groundbreaking science that we have, but again, capture and count doesn't tell you the whole story. So where one cell comes in is a groundbreaking science to isolate the captured cell into a live single cell. Okay. So traditional circulating tumor cell CTC, the endpoint was capture and count and give the report. For us, this is not an endpoint, this is the starting point. Okay. We begin with this captured and isolated live single cell, and we do the imaging modality on it, we do the NGS on it, we do the multi-omics analysis on it, right? And so we have a much deeper insights that enable the doctors to say what's really going on. Is the therapy working? If it is working, why it's working? If it is not working, why it is not working? And what could you do potentially to change the course of the therapy? Can we add, can we increase the dose? Can we add the combination therapy? Can we change something? Or if nothing, if it is working, can I reduce the dose level? Can I give those chemo holidays? Can I, do the de-escalation, right? So all these questions are unanswered in the minds of the oncologist. Okay. We provide AI assistant to the oncologist, okay, based on this scientific data that is presented, multi-omics, data that is presented for a given patient at a given time point, to be able to provide them meaningful, actionable information so that
Mhm.
they can change the course of the treatment, personalize the treatment for a given patient, which will
Yes.
improve the outcome.
Yes, and cancer sometimes change as well, right? So you have the mutations. Some mutations take over versus the other, right? So in that sense, like if it is if it's growing, like you can detect like, okay, what is the new mutation that's coming up, right? And then the ecologist can then change the treatment accordingly as well.
It evolves, okay?
Yes.
And so many successful companies, and I'll give you one example, Natera. It does the tissue at this baseline, at the starting point. It hones in on an information about 16 gene panel, and it only monitors those 16 genes longitudinally. Now, so one of the most successful company, one of the most successful test, but it does not address the evolution of cancer. Okay. You cannot take a snapshot. It's like, I've taken, before leaving my home, going to the office, if I take, let's say, Google Maps, right? And I take a snapshot and I start driving. And if there is an accident and, I have to do the stuff, I can't just, it cannot be a static thing. Cancer evolves, so you have to, the testing has to kind of adapt to the evolution of the cancer, and I think the newer sciences, newer tests, newer diagnostics is really more reflective of the evolution of cancer, more adaptive to the evolution of cancer, and provide the tools to the oncologist, that reflects the changes that happen. That's how
Yeah.
the outcomes are better.
Yeah. So this Onco Insights, right, what you described as the clinical diagnostic. So it's a clear lab model. Can you talk a bit about that? How
Sure.
did you get like certified, and what would does that entail?
Yeah. Good question. There are two ways a diagnostic company can offer, the new innovation. Okay. More traditional way is basically, your invention, you go through the IP protection. Then you go through a regulatory pathways, which is the 510(k) for an FDA clearance, right? So you find a predicate device, some similar test that has already been approved by FDA, and you establish that your test is as good, if not better, right? And you do that. Now, that takes a long time, 3 to 4 years, and many tens of millions of dollars before you see your first, revenues, etc. The other way is a provision called LDT, lab developed test. So if you have a lab that is CLIA and CAP certified, CLIA is a regulatory body, that does the inspection of the labs, ensuring that they follow proper protocols. It is run by a very qualified, board certified pathologist. Okay. And if you establish and prove that you have developed a test that has a good science backing it up, good publication, you have done a good, scientific experiments, quality control, and all of that stuff, then, on a limited basis for a limited target, you could offer this, lab. Okay. So we have adopted a two-prong approach, a two-phase strategy. We have a CLIA CAP certified lab in Foster City here in Bay Area, okay, where we could take the samples from the patients. We process the sample and produce the results and the reports and give it back to the oncologist, based on which they can basically, take the actions. Our initial focus is not commercial diagnostics. Our initial focus is to provide these services to pharma and biotech companies that are doing clinical studies, clinical trials. Okay. So, pharma companies are always pushing the envelope. They want newer technology. They want new science to work, right? And so it's a shorter path, helping them accelerate their translation research, their clinical trials, gives us a shorter path to revenue, and gives us an add-on confidence that our test now then can become a companion diagnostic test and
Yes.
eventually a commercial diagnostic test.
Okay. Okay.
Yep.
And so tell us about like the information that you provide to the clinicians or to the clinical trial coordinators, right? Is that about, okay, well, there's these mutations, or like what kind of information are you providing to them?
Our Onco Insight, which is the name of our assay, is an LDT, right? So we get 2 tubes of 10 ml blood each. We provide the collection kits. They are available at the doctor's office. Okay. A phlebotomist goes, with the patient, we draw the sample, and those samples come to us. Okay. It's a 1 day turnaround time. It might take 2 days depending on the location, etc. And then we do the processing of the samples, right? So what do we do? It's a 2-track. The first track is we extract the DNA, from the blood sample. And we have developed, part of Onco Insights is a ctDNA section of the test, where we have developed a very novel 1080 gene panel that works on the liquid biopsy. and it produces the results. It tells, okay, what are the what are the genes, what are the mutations, indels, fusions, very comprehensive stuff that we provide. It also provides the meaningful interpretation, not just the analysis of what genes and mutations are present, but we have created a back-end informatics comprehensive knowledge base, okay, which allows us to provide actionable reporting. Okay. And so we characterize based on NCCN guidelines, okay, that what is the level 1 evidence, level 2 evidence, level 3 evidence, right? So very comprehensive stuff that has the actionability because of the presence of these genes or these mutations or these translocations or these things. We are able to say that most likely this is the recommendation of these drugs will work, these drugs won't work, etc. It's a very actionable report that oncologists can use it as a reference. Ultimate call is made by the oncologist. We only analyze and interpret and present the data. Based on that data, oncologist will make a call and do the stuff, right? So the onus, that responsibility is with the oncologist. We are merely the tools to provide the information to the oncologist. The second cue, sorry, please go ahead. Okay. So this is the DNA side of the story. The second tube, then we extract, we capture the circulating tumor cells. Okay. And then we extract the DNA and the RNA, okay, and the proteins. And then we run our single cell assay and get the full analysis of what are the DNAs on the each single cell. And usually a 10 ml blood will give us somewhere between 5 to 15 circulating tumor cells. That's 1 in a billion, right? And so we are now honing around somewhere 10 cells, right? So if you get 10 cells, each single cell, okay, we do the whole genome, the whole transcriptome, and a targeted proteome, so about 20 proteins, right? And we provide a very comprehensive report of what genes, what mutations, what translocations, what fusions, what proteins, right? All of that stuff. Now, this in conjunction with the ctDNA, right? So our onco insights, we are not saying CTC is better than ctDNA or ctDNA is better than CTCs. What we are saying is that when you combine the two and give an integrated report, that is much better than any individual, technology. Historically, ctDNA or cell-free DNA based tests have shown that 18 to 19% of the time, you have NMDs, no mutation detected. Okay. So even if the best companies, the Garden, the Natera, the Foundation, the Tempuses of the world, this is the industry average. Okay. So when you combine that with the CTC together, that thing, the sensitivity goes from, 70, 80% to high 99, 90, 90s.
Oh, wow.
Okay. Because
Yeah.
what is found in ctDNA or what is missed in ctDNA, CTCs capture it.
Yes. And
Yeah.
and this is like more like usually we try to do like, there's multi-omics, right? Because you have different different parts of the overall cell you're capturing, but now you're adding another very new angle on top of the multi-omics. You're adding the CTC angle as well. So okay. Yeah, that's great. And so this rich information that you're providing. So let's talk about like both the settings, right? Like the one is in more like the commercial setting, and then the other one is in the clinical trial setting. I said clinical trial setting. There's a lot going on right now. So so based on this, what kind of decisions can be made
Mhm.
about the clinical trial, right? Because this patient is there to get a specific therapy. So now you come up with this. So now what changes? Because either the patient is taking that therapy or they're not, right? So how does this change that?
Very good point. There are two separate tracks here, right? One is the clinical diagnostics, which we are doing it in India, okay, where the doctor wants to know what is the right therapy for a right patient at the right time point and the right dose, right? So we provide them a full comprehensive report to help him or her make those decisions, okay? That's one. The second part of that workflow, the clinical workflow, is okay, you have given the drug. Okay. Uh, Now, is the drug working? So, therapy response monitoring is another aspect of it, right? And there are two pieces to it. One is the MRD, the minimum residual disease, right? 95% of the time you have taken care of. But that little 5%, right, that becomes a problem down the road, right? And so, how to handle that, right? So, MRD and the therapy response monitoring, that's the clinical side. Now, you ask me the second question, how is this Onco Insight assay helpful in the clinical trial setting for the pharma? Okay. So, I would say 3 or 4 key points that we address. First and foremost, when you are doing the screening for patient recruitment, is this the right patient? Usually, you have inclusion criteria, but most of the inclusion criteria are very high level.
Yeah.
Breast cancer patient, women between the
Yeah.
age of this and this, you know,
Yeah. It's not going at the molecular level at all, right? Or the
not
cellular
going
level.
right,
Yeah.
right.
Yeah.
So that's why even when you recruit patients, they don't perform very well, right? So by leveraging assays, tests like Onco Insights, a molecular level, you have a much better fit. So patient selection is a very interesting area where we can help the clinical trials be successful by recruiting the right patients. Equally important, patient deselection. Okay. How not to recruit the wrong patients, because one bad apple messes up the whole fruit basket. Okay. Right. And so, again, when you integrate the ctDNA and CTCs, you have a much richer set. So, we could basically find better patients, recruit the right patient, okay, and deselect the wrong patient. So, that's one, clear value proposition to the pharma companies that are doing biomarker based clinical trials, okay. And we know biomarker based trials have at least 2 times higher probability of success from the FDA clearance. Okay. But typical biomarkers that were based on tissue was very hard, right? Because then you have to have tissue biopsy. So cell biopsy or a liquid biopsy is relatively easy because you just take the blood sample. So it costs less and it's much easier to monitor and manage, right? So that's one.
Yeah. And it's also faster, most probably,
It's
right?
much faster, right? Much,
Okay.
much faster, right? That's one. The second thing is, okay, you have now recruited the patient, you started, you have dosed the patient, right? Is it working or not working? In the today's point, right? Let's say if you're doing immunocancer therapy, whether my therapy is working or not working, I have to wait for 3 to 4 months and I have to go expose the patient to either a CT scan or an MRI or a PET. And then I would know using what they call it as a RECIST criteria, okay, whether the tumor is shrinking, whether it is not shrinking, or whether it is growing. But look at the time frame now. For every patient under study, I have to wait for 3, 4 months before I know whether my therapy is working or not working. Okay. Let's say you're targeting the drug with Herceptin, HER2. Okay. And you've given the drug and now you have to wait. Now, instead of that, 3 to 4 months wait, within a matter of couple of weeks, okay, if I could tell you whether the drug is working or not working,
Yeah.
wouldn't that be a great value?
Yeah. I mean,
Right?
yeah, that
And
was
how
probably
do we do
fantastic. Yeah.
that today? Okay, by capturing circulating tumor cells, right? First of all, if the drug is really working, the tumor cells should really go down.
Yes.
The ctDNA should really go down,
Yes.
right? Okay. But you have an 18-19% of NMDs in ctDNA, right? So you miss out those 20%, right? But if I can capture CTCs, right? And if I say, oh, I have CTCs where HER2 is still expressed on the surface of the CTCs, that means my therapy is not quite working. Okay. And then by doing this multi-omics analytics, if I could say, what is the reason why it is not working? Okay. And again, I am making it up, but if there was a marker, let's say EGFR. So if there's an EGFR positive marker, then we know historically that HER2 will not work. Okay. So you don't have to wait for 3-4 months. You can make those critical decisions now, and you say, based on multi-omics profile, okay, what ails combination therapy, or can we switch over? Can we change something, right? Or can we add something to it, right? A combination therapy to have a resistance prediction, right? If you can predict the resistance, then actionable information will help you add some combo therapy, okay, anticipating the resistance so that the outcomes would be better. So you see the combination of information, multi-omics data from a CTCs or a single cell, coupled with AI to do the prediction, is going to transform this field and make the clinical trials much more successful. Okay, because you're taking actions proactively. You're not
Yeah.
waiting for 3 months, then realizing that, oh, my drug didn't work. That's a loss of time and loss of money.
Yeah, and it's also the tumor has progressed by that time, right?
And by that time, tumor has progressed, right?
Yeah. Yeah, and so you're like, this is early detection of like the tumor increasing or tumor changing, right? So this is I mean, this will be adding a lot of value. So how are you thinking about like how can this be commercialized? Like we talked about the clinical trial study, but in terms of like the clinicians using that in the US and in terms of how you are thinking about commercializing, would there be any like payer barriers or so? So what is your plan around that?
Currently, the way we are taking is help the biopharma companies. And if they use our assay to accelerate their translation research, then one path would be this assay becomes a companion diagnostics. That with that drug, right? So that's one path we are pursuing. Parallelly, we have initiated number of clinical studies for specific utilities. Okay. And once we get the 510Ks, for instance, right? Then we'll do the MolDX route, okay, to get the reimbursement, right? Once we get the reimbursement, then we'll make this commercially available, okay? And we have two choices. Either we build our own, sales force and the whole operation, or we partner with larger established labs who already have the partnerships in place, okay? And currently, what our interest is, our focus is going to be, we are the R&D. We can come up with the new assets. And our intent is to leverage strategic partnerships for commercialization of our assays. In the commercial setting, we will probably strategically partner with other larger lab setups.
Yes, that makes total sense. And in terms of like we talked about the fantastic science and technology around it. So how do you measure the sensitivity and is there like a lot of variation? Like how have you can you talk a bit about that?
We go through what we call the analytical validation study, okay, where the sensitivity, the specificity, the repeatability, all those factors are well covered, okay. We have on our team member, our VP of clinical development, comes with years of experience doing it with the drug companies. So very regimented, very strict, very stringent, criterias that we have brought into our practice. So we do the analytical validation, then we do the clinical validation before the assay is ready for putting it into the commercial stuff, right? Where we do that. We also do it independently with third party independent labs. So we were very fortunate to have a collaborator like the University of Augusta, Dr. Cole, who has been doing this kind of stuff for companies such as Agilent, Illumina, and many other well-established companies have, taken their help to do the analytical validation. We did one study where we compared our assay with an FDA cleared assay. It's called PGDX, the company which has been acquired by LabCorp. Okay, it's one of the very popular, successful, well-established assay. And I'm very happy to report to you that we showed 99.8% equivalency, equivalent to an FDA cleared assay done completely independently, you know, at a lab, which is the University of Augusta. And that paper will be published very soon. Manuscript is ready. We are working through the final details. But that
Right.
gives us the confidence that independent lab was
Mhm.
able to conduct an independent study with an FDA cleared assay, and we
Mhm.
show a substantial equivalency. So
Mhm.
that becomes interesting.
That's amazing. Congratulations on that.
Thank you.
Mm.
Thank you.
Oh, and so now, as we talked about, you talked about this third generation, right? Where is this field going? Like, let's say 3, 4, 5 years later, you're the ongoing so that will be on the market. What will be next? Like, where is this field going? What do you see as like the next, like the fourth generation in this area?
Great question, and that's the reason why we are excited. Our mission is to democratize precision oncology, making it actionable, making it accessible, so easily accessible across the country and around the world, and making it affordable. Okay. So that's our vision, that's our mission. And we believe that cell biopsy coupled with AI is the transformer. Okay. Uh, our ultimate goal is can this be a simple assay with a simple blood draw be able to detect way early, and I wouldn't say displace completely, but significantly reduce the need to wait for 3-4 months. Okay. And look at the sensitivity of CT scan. Unless the secondary tumor grows to 9 to 10 millimeter, you can't detect it. Okay. A 9 millimeter tumor means billions of cells. And here I am looking at single cell. Okay. So if we could utilize good AI to analyze the data and show the equivalency, substantial equivalency, okay, don't have to wait for 3-4 months, don't have to spend, thousands of dollars. It will transform the care. And particularly for survivors, cancer survivors, the people in the remission, where they have to go through once a year, twice a year, once a quarter, and go through the agonies and the pains, spend a lot of money, don't have the sensitivity. So the celebration that my pet is clean is not very long lasting, right? Because it didn't get detected because your tumor wasn't quite 9 millimeter. But within a month after you celebrate your cleanliness, you might come back again. Okay. So early prediction of a reoccurrence, if I could do through cell biopsy, okay, then I think we have conquered the big time. 90% of the cancer deaths happens because of the reoccurrence, right? And, reoccurrence, the root cause of it is the CTC, right? Because the metastasis, right? And if you can capture early and create a sensitive, specific assay, which is minimally invasive, a simple blood draw,
Yes.
I
Yes.
think it will change the field, and that's our mission. And our
Mhm.
goal in life is to impact at least 1 million cancer patients' lives across the country and around the world.
That is fantastic. And all our best wishes to you about those.
Thanks.
Um, Mohan Uttarwar, co-founder and CEO of OneCell.ai. Mohan, thank you very much for your time today.
Thank you very much, Amar and the team.
Well, that was a great conversation. What did you think?
Okay, it was fascinating to learn about the progress being done in this area of diagnostics, liquid biopsies, right? So we've been living through this when it was tissue biopsies, and then liquid biopsies came in, and now it's liquid biopsies, but only single cell, and the precision that we can have now from scientific and technology point of view of really focusing on one cell and then really figuring out from there what could be what's happening with the tumors. It's just fascinating how we're rapidly advancing the science and technology and bring which is really at the forefront of healthcare at this point.
It must be 20 years now that liquid biopsies kind of first emerged on the scene, and there was a lot of excitement early on about their potential, but the impact has been limited so far. To what extent do you think what OneCell is doing addresses the existing limitations of liquid biopsies?
So they are not only using just the liquid biopsy and then doing um, some regular experiments. I mean, they are doing a lot of the multi-omics on this. So he talked about genomics, transcriptomics, proteomics, as well as a lot of stuff about understanding the morphology of the cell. So they're bringing in a lot of different types of analytics together using AI, using analytics. So, and they're also, he talked about gene assay, right? About over 1,000 genes in that. So it's really going deep into quite a bit understanding much more about the cell, much more about the DNA, but also about the protein. So it provides a lot more information. It's also, we have to think about. What he talked about was the report that they generate, which is not just about okay, well, this gene here, and then but then they are using NCCN guidelines to then connect this to the drug therapies, and then really talking about okay, well, what are really the actionable mutations that we can see, because that's the biggest problem, right? Okay, there's some mutations that you can detect in any DNA, but so what? What do you do with that? There's no drug for that, right? So now detecting these actionable mutations, but also tying them to the therapeutic options, I think that's going to be great. And then it's also, as we get more and more therapies, this is going to even going to be more, all right, because they're going to be able to say, okay, well, these were mutations that were not necessarily actionable before, but they will be now, right? So I think that's definitely very, very good. And also, I mean, to me, the big thing is to be able to detect early, right? I mean, like he talked about 3 to 4 months. That's a huge amount of time in a cancer patient's life after diagnosis. So getting any of this information before you see that, like the tissue growing or so, I think that will be that'll be definitely a big step forward in terms of changing the therapies for the patients, and it should result in better lifespan, higher quality of life in the long run for the patients.
Mohan said this combination of applying a multi-omics approach and cell analysis to circulating tumor cells wouldn't be possible without AI. Is this a good example for the potential of AI to change clinical practice and allow us to finally realize a long-held vision of personalized and precision care?
I mean, anytime you're using omics, you have to use AI because the omics data is huge. I mean, you talk about a single genome, that's huge. You're talking about proteomics, you're looking at like all the proteins in the cell, right? So you definitely need AI and advanced analytics to process any omics data. And then as you're looking at combining those data, combining different types of omics, but also combining a lot of this cell data, you need the AI to glue all of that together. So yeah, you have to have it. And of course, in diagnostics, I definitely think that AI has a big usage in diagnostics because you're trying to figure out what's happening in this cell, and the cell has way too many components that you do need AI to do the analysis for that.
The initial focus of OneCell is on helping biopharma companies use the technology to improve their drug development efforts and help build a case for its test as a companion diagnostic. What do you think of that strategy?
It's a good strategy. I think they are following both strategies, right? Like going with the 510K route as well as this. But here, I would say definitely this gives them a lot of experience about like how it works with in the clinical trial setting with how the clinicians will use this information. But also the big, I would say the big advantage there is to be able to have it as companion diagnostic. So if they can get approved as companion diagnostic for any drug, then that the test needs to be used, has to be used whenever that drug is to be prescribed, right? So that I think will be fantastic for them commercially, and that also is just going to help in getting the general usage application. I mean, they will be generating a lot of data that will be helping with those applications as well.
As you think about the various stakeholders around clinical practice, providers, payers, regulators, drug companies, and patients, what do you think about the potential for AI to become integrated into care, and what will it take to win acceptance for doing so with these different stakeholders?
The way I think was that the physicians and patients are always very interested in understanding what is happening. Regulators it's going to be more about, okay, well, they're doing the trials. It's about the insurers and whether they are going to reimburse this or not. I would say the one of the things is going to be, yeah, whenever you talk about AI, how expensive is it? When you're talking about next generation sequencing, doing a lot of the proteomics analysis, that is, it's gonna take some money, right, to do all of that. So, but then what they have to think about is that the benefits from here, right? So, at the level for the patients, but also at the population level, is this going to really then help prevent the patient from recurring? Is that going to help? Is it going to help in increasing the quantity of life, but also the quality of life? Is that going to help in maybe less therapies later on because you're able to and the wrong therapies later on, right? Because you're detecting very early on what should be the right therapies. I think those are some of the advantages that the payers will take into account, and then they will do the analysis. But I would say that it's gonna. I mean, there's definitely the potential to have this in clinical practice. I think at some point we have to get there, right? Like we have to have these diagnostics, the early diagnostics, to be able to detect cancer early on, or like the changes in cancer. Because I mean, I always think about like diagnosis is much better than cure, right? It's you're always much better off with diagnostics than cure, and something like diagnostics like that, it is going to require AI because cancer can like there are so many genes that can be potentially involved in cancer that you need to have analytics. It's not going to be just one test that is going to be able to detect what the molecular mutation is. You are going to need analytics and AI to be able to detect holistically for the right therapy.
It was exciting to hear, and a great example of how AI can not only change drug development but clinical practice. So, Amar, thanks as always.
Thank you.
Thanks again to our sponsor, Agilisium Labs. Life Sciences DNA is a bi-monthly podcast produced by the Levine Media Group with production support from Fullview Media. Be sure to follow us on your preferred podcast platform. Music for this podcast is provided courtesy of the Jonah Levine Collective. We'd love to hear from you. Pop us a note at danny@levinemediagroup.com. For Life Sciences DNA, I'm Daniel Levine. Thanks for joining us.